Assalamualaikum, welcome back to BioCore. In the previous lecture, we learned that enzymes speed up reactions and are essential for life. But here is an interesting question, that is, why does one enzyme only work on one specific substance? Why can't an enzyme work on everything? Today we will understand enzyme specificity, active sites, lock and key hypothesis, enzyme-substrate complex, and naming of enzymes. So let's begin. Firstly, we'll discuss what is enzyme specificity. Specificity means that each enzymes work on only one specific substrate. It doesn't work on another substance. For example, an enzyme that breaks down starch will not break down proteins. An enzyme for protein will not act on the fats. Enzymes are highly specific due to their shape. This point is very important, that enzymes are specific due to their shapes, and you have to keep it for your exam point. Now discuss an important point, active sites. The active site, it is a very important part of enzymes. Each enzyme has a specific and special region called the active site. This is the place where the substrate binds, the reaction take place. The shape of the active site is very specific. Now discuss the very important model, that is lock and key hypothesis. To explain enzyme specificity, scientists proposed a model called the lock and key hypothesis. According to that hypothesis is that the enzymes is like a lock enzyme and the substrate is like a key. So only a correct key can fit into the lock. This means only the correct substance can fit into the enzyme's active sites. So this is the very important point. Now we discuss the important point, that is the naming of the enzymes. Now let's look at how enzymes are named. Most enzymes named end with ASE, like the amylase acts on the and break the starch and it acts on the starch. Protease, it helps to the breakdown of the proteins. Lipase, that helps in the breakdown of the fats. Sometimes enzymes are named after the substrates and the naming and the type of reactions. Now we discuss the enzyme-substrate complex. When the substrate fits into the active sites, a temporary structure is fit called the enzyme-substrate complex. Now discuss its step. Enzyme bind to active sites. Enzyme-substrate complex forms. Reaction occurs. Product is formed. And then product leaves. Enzyme remain unchanged, and this process happens very quickly. So you have to keep that points in your mind. Now we are going to discuss that the speed of enzymes reaction. Reactions can actually happen without enzymes, but they would be extremely slow, taking hours, days, or even years. With enzymes, the same reaction happens in seconds. For example, the enzyme catalase can break down hydrogen peroxide very quickly, thousands of molecules per second. Now here is the everyday example, that is about the amylase. You have to try this simple experiment. It is like you have to chew a piece of bread and keep it in your mouth. After some time it starts tasting sweet. Why? Because the enzymes amylase in saliva break down starch into sugar, and the sugar, that is the maltose, and this causes the to taste sweet. Here is the point, that is the very important point, reaction in stages. Many reactions happen in stages or steps. For example, if it is the breakdown of the starch in the first stage breaks into maltose, and in the second stage it is converted into glucose. This is a very important point about the stages and the steps. Now we discuss the specificity of enzyme. Let's understand this with an example. An enzyme that breaks starch into maltose will only work on starch, will not work on proteins or fats, and this shows how specific enzymes are. and the point is that enzymes can be reused after the reaction. The products leave the enzymes is not used up, and it can be used again and again. That's why enzymes are needed in a very small amount. Let's quickly revise today's key points. That is, enzymes are specific. Each enzymes work on one substrate. Active sites is where substrate binds. Log and key models explain specificity. And an enzyme substrate complex forms temporarily. Enzymes are reusable. Reactions can occur in stages. Reactions and the enzymes names usually end with A's. If you found this lecture helpful, like the video, share the video, and subscribe to BioCore for complete biology lecture series. See you in the next lecture. Till then, take care. Allah Hafiz.